So this slide is labeled epididymis human section. That's what it's labeled. But it has not only epididymis, but testes in it as well. So it'll be good for us to use it for everything else. So focus. All right, so that is testes there. And then you've got this big thick stroma, this big thick wall of connective tissue. And then down here, you've got epididymis. So, let's talk about the differences in what we're looking at. So on this one, the first page, it's got just kind of an overview of what's going on the testes, right? So first things first, you've got the big thick capsule. Look at that thing. That's huge, right? Big thick capsule, right? So that's your tunica albuginea, right? Your big thick tunica albuginea there. This is seminiferous tubules here, right? You can see them easily. And then blood vessels. Well, where's blood vessels? Let's see. I saw one. There's a nice big one there. There's a big blood vessel there, right? Can't miss it. Looks very different. Everybody see it? Anybody not see it? And what kind of blood vessel is that? It's an artery. Very good. What else is there? Is there more than one? There are two of them there, right? See them now? There's a big one and a small one, right? Anybody want to make a guess at what kind of arteries those are? What are the three types of arteries? What's the one? What's an aorta? What kind of artery is that? Elastic, right? Then muscular and arteriole, right? So what's the bigger one? Muscular. What's the littler one? Arteriole. Very good. All right. What's next? That was easy. Now the actual functional part, right? So if we look at this, see if I can get my color in my. All right. It looks like a big hairy mess, doesn't it? Kind of. So what are we looking at? What's the first thing on here? Tunica propria, myoid cells, teratoid cells. Yeah. Okay. So this is pretty much all the stuff, although number 7 is kind of weird there. I wonder why I cut off 7. You know what 7's supposed to be?
Spermatid.
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 Spermatid, not spermatid. Spermatid. All right. So now we have to look closer, right? So let's pick one of these and zoom in a little bit. I don't really care which one we pick. Pick one, right? This isn't bad. This isn't bad at all. Okay, so I didn't bring my pointer thing. Is it sitting on my desk? You know where my office is, right? Tell my secretary I want my pointer thing off my desk. And tell her to give it to you. All right. So the first thing we have to look at is the connective tissue that goes around the seminiferous tubule, right? Which is the tunica propria. That's what that pink band is that goes around it, right? The tunica propria. if you look inside the tunica propria, you can see some nuclei, right? Do you see the nuclei there? There's one over there, right? See the little nuclei in there? It's locked. Thank you. All right. Now, as I was saying, let's go back to this here. So, tunica propria is this connective tissue capsule around these, like this nuclei here and this nuclei here. Those are the nuclei of the myoid cells. Myoid cells are what maintain and manage the tunica propria, that connective tissue capsule around the seminiferous tubules, right? How to think about it. So remember that the seminiferous tubule is a tortuous thing, it goes all over the place, right? I always kind of think of, I don't know if you know how to do this. Have you ever seen, you know, everybody knows what a sausage is, right? How do they make them? How do you make a sausage?
Grind the meat and stuff.
You grind up the meat, put your seasonings in it, whatever they are, right? And then you stuff this tube thing, this tube, and what it's made out of is dependent on other things, right? What they used to do in the old days, you might know what it used to be? It used to be intestinal lining of whatever organism, right? It's still biologic in nature. I don't know if you know that or not. But anyway, so I always think of kind of that as this, right? Think of this tube as a sausage kind of, right? And so this is that wrapper around the outside of the sausage, right? And then you have your working parts on the inside. All right. So Sertoli cell, we talked about Sertoli cell already in the class, right? Sertoli cell is the big important part that does all the functions, right? Remember I said it's pyramid shaped, right? Pyramidal. So where do you see a pyramid shaped cell in here? There's several. So let's start with the one that's most distinctive that you can see, right? Where is it?
Left.
There. Look at that. See how big that is? That's one. It's easy to see there. You can see the cytoplasm go up into that big, long, kind of pyramid shaped. It's not true pyramid shape, but close, right? So that's one. The other thing that is a key thing here is that the nucleus of a Sertoli cell is really big. So the other thing you can do is you can look at like this one and this one and this one here and this one and that one, and those are all Sertoli cells. So you can just look for that big nucleus, and that usually is the answer. Too small. It's probably the lowest one on the left, to be fair. I should have. Hang on a minute, I'm looking. Sorry, I'm looking for something. Hold on. I'm trying to see if I have something else here. The things I have and the things I don't have. All right. So there's a different stain, right? This is the red stain, right? Anybody know what stain this is? Do you think it's H&E, or is it something else? Looks pretty close to H&E, right? not a lot of H, right? A lot of E, right? And that's not gonna work, that's oil, okay. This is as high as I can go without oil. So let's look at this area right here and talk about how weird it is, right? So if this is a Sertoli cell, right? What are these?
Primary spermatocytes.
Those are spermatogonia,
Spermatogonia.
right? They're down here connected to that connective tissue down there. Remember how I said they were connected to the connective tissue and thereby maintain that connection and stay there, right? And what happens when they divide, right? Spermatogonia divide mitotically, right? And you end up with another spermatogonia and what?
Spermatocyte.
Primary spermatocyte, right? And then what happens is that primary spermatocyte then undergoes a meiotic division and changes in shape and size. So let's see, let me see if I can find a good example of the first division and then the second division. How about that? That's not bad at all. Let's see that. Okay. I like that. That's pretty good. All right. How's that look? All right. Let's look right here. These are spermatogonia. And this cell are primary spermatocytes. So they're usually right by each other, one above the other, right? One still connected to this connective tissue, one not connected to the connective tissue. So then this cell will divide mitotically forever, right? This one will now become meiotic and do meiotic divisions. The second division, which is a meiotic 1 division, right? The first meiotic division, ends up with cells that are about this size. So these are all the result of the first meiotic division, which are secondary spermatocytes, right? The last division, which is the second meiotic division, is what produces the spermatids. Spermatids are relatively small. Think these things, little tiny things. They're not very big at all. Here's some here. These are really good examples. These little dark things that you're seeing. These are all spermatids. Those little dark things. So the nucleus gets very, very compacted in a spermatid. It's just very small package. What is the other thing that's occurring that's very weird here that I haven't talked very much about in here, but you would talk a lot about in embryology, you talk a lot about it in basic biology a lot. What happens in male gamete production that doesn't happen in female gamete production?
Consistently?
When you're dividing the cells in making sperm, you have equal divisions of cytoplasm, so they all get smaller and smaller and smaller with each division. Because it's rapid division, you don't have time to replenish the cytoplasm. So the cells get smaller and smaller and smaller in male reproductive gametes. So with each division, they get smaller, which is a good thing to remember, right? In egg production, what happens though? We haven't gotten there yet, but what happens in egg production? And we're not going to talk about it very much in here. It's not going to be anything that we care about really in here because it's not pertinent to what we're doing. But, so, for example, you start with one cell, go through 2 meiotic divisions, you end up in sperm production with 4 sperm, right? In egg production, what happens? You
You
start
only-
with one cell,
You have one cell at the end.
you end up with one cell at the end and
Yeah.
3 what are called polar bodies, right? So 99% of the cytoplasm stays with one cell in the female reproductive system, right? So you have one cell that's still large and the other 3 are nothing other than basically throwaway material, right? So the thing to remember here is that they get smaller and smaller and smaller with each division. Any questions on any of that? I'm going to try to find one of the purple slides on this and show you some more on that.
Where was the secondary spermatocyte again?
So secondaries are kind of this intermediate size, right?
Mm-hmm.
So your primaries are going to be the same size as this. So same size. These big ones here
Yeah.
are primaries, right?
Mhm.
These that are kind of medium in size, like this whole cluster here, are secondaries,
Okay.
right? Your spermatids are the little tiny dark specks that you see, because it's almost all nucleus and not much else, right?
Thank you.
Could you, which ones are the myoid again?
Myoid cells are in the connective tissue capsule, right?
Okay.
Some of these are better than others. This isn't a bad one here. It's kind of good. It's very busy, right? How about that? It's very busy. There's lots and lots of cells doing. What's the thing that you take away from this, right? Let me go back to this here, like this. What do you take away from this? How active is this thing? Remember how many sperm are produced in the male reproductive system at any one time. First off, it's always working, unlike the female reproductive system, right? It's always working. It's always putting out large numbers, right? So the way to think about reproductive differences in males and females is males are like shotguns, right? Let's put out as much as we can and hope we hit the target, right? The other part is it takes lots of sperm to even cause one sperm to be able to fertilize the egg, right? Female reproductive system puts a lot of energy into just a very few eggs, right? Yeah.
I think also for males, they have a lot of genetic issues with the sperm.
Anytime you do meiosis, you're gonna get combinations that don't work, right? I mean, we could talk about viability or reproductive success, but I usually talk about that in embryology, but that's easy enough to talk about, right? Who can tell me, let's say you have sperm and egg get together, and in the right quantities, in the right environment, right? No matter, everything's perfect, right? You have sperm and egg get together, how many times is that gonna result in a successful fertilization event? We reproduce easy, right? So it must be very common, right? You would agree with that. So I'm talking about everything else. The sperm got to the egg. There's no issues in pH. Everything's working great. It's just a matter of the two joining together. How often does it happen? You would think it'd be great, right? What's the percentage? About 30%. That's just fertilization. That's not implantation, that's not development, that's not nothing else. That's just fertilization, right? 30%. And that's if everything worked perfectly. It's not as common as you think. So what do we do to compensate? What do humans do to compensate for the lack of success?
You just try a lot.
Try a lot, right? If you don't get it right the first time, continue until you do, right? And we know what that's called, right?
Insanity.
What did you say?
Insanity. Doing the same thing over and over and over, expecting a different result.
Sex is insanity.
Some people would call it that, yeah. There's a reason why we have a sex drive, right? There's a reason why that is in every species out there. Two prime characteristics of life, right? One of them is reproduction, right? I don't have to stress this to you, you know what I'm talking about. It's not anything new. All right. Anything else about this before we go on to something else? All right. Now we're going to look at the interstitial cells, right? So now we need to look somewhere else, right? So let's look, where's a nice example? So let me go down a little bit. So you start looking at this, let me go down some more. You start looking at this and you think there's a lot of spaces that are interstitial, right? Would everybody agree that there's a lot of interstitial space? What's all this? Is that all interstitial space? Well, some of it is, right? What's this? Some people would say that's a patch of interstitial cells, but that's not what that is. What is that? That's where it's just barely cut on the edge of a seminiferous tubule. So this is actually seminiferous tubule, this is seminiferous tubule, this is a blood vessel, right? This is nerve tissue here. There's other things in there, right? So don't be thinking that just because it's not in a seminiferous tubule that it's not going to be something that's not an interstitial cell. I don't know, that didn't come out right, but you get what I mean. So you kind of have to hunt for what you're looking for. And they do have a fairly characteristic look to them, which is good because they are different looking than the other cells, right? Let me zoom in a little bit and see if I can find one. It's kind of two of them, but that's all right, that'll work. All right. Anybody see them? Where's the cell that looks weird? That's one cell, that's another cell. There's two of them side by side. It's a little odd looking, but that's two of them side by side. Bright red cytoplasm, nice dark nucleus. Fairly distinctive looking. They look different than all the other cells, right? Look at these cells. These are just connective tissue cells, right? This is lymphatic here, this is lymphatic there, right? Yeah, those big cells there, okay? That's your cells of Leydig, right?
The cells of Leydig produce testosterone, right?
That's correct.
So how did the testosterone get to the or get to the rest of the body? I don't see very many capillaries or blood vessels in it.
There's no capillaries in the seminiferous tubules. You don't want blood in the seminiferous tubules.
That's right. That's right. So I guess with that like blood testis barrier, how does the testosterone cross that?
It has to do with the myoid cells, actually. The myoid cells actually move it across that membrane. Okay, let's go on to another structure. I will see maybe so after Thanksgiving, we're gonna have Monday, Tuesday, Wednesday, we're gonna be doing presentations those days, right? That's the week we're doing the presentations. Let me look at my schedule. So sometime Monday, Tuesday, or Wednesday, I'll try to figure out a time when I can slot off an hour or two. I will try to schedule some time to come in here and do like a review session. Oh, let me look at my schedule, see what I can find out. But don't let me forget that. But I'll try to schedule a review session for that Monday, Tuesday, or Wednesday before the last lab practice to do a review. Okay? All right. Let's see, let's move on to epididymis down here, right? Now, some people would say that it looks the same, right? And the answer, it doesn't, right? It doesn't look anything like it, right? Remember structure, color doesn't matter. Remember structure. What do you see that's different? It's very different, right? Well, first off, the area, so this is the epididymis, it's a tube as well, it's got the same tortuous path, so that's the same, right? But look at the interstitial area, it's much different, right? The area between the tube is very different. And secondly, the area inside the tube is very different. So the outside the tube thing, it's mainly connective tissue, it's almost solid, right? There's no gaps in it like the other one does. Not quite as many blood vessels, not as many other cells in there and other things like that. It's just mainly connective tissue in there, right? A few blood vessels, not much else. But if you get into the inside of the tube, you'll see very quickly that it's very different, right? That doesn't look anything like testes, right? You'd agree with that, I hope? What does it look like? It looks like almost like grass or a brush border or something like that, right? What is this? It's epithelium. This is very much think intestine here. It's got these little villi that go up and down and up and down and up and down, right? The stereocilia, these long microvilli, are actually on the end of it. Let me see if I can find some and show you. And let's see. Oh, here we are. We can see them quite well here. Let's see. How about right there? That's a really good shot. I can go up one more time. All right. This and this, they're very wispy, just long little stringy things that are sticking up. This isn't bad, but you can't really see all of it there either. If you focus in and out, you can see a little bit more of it. It actually goes it further than that. But those are the stereocilia, those long extensions there that go up into that area. So this is your epithelium, that's stereocilia there. What's this stuff out here? Free spermatozoa. Loose in the lumen, right? Lumen is the empty space in the middle, right? There are different stains that look much different than this. The picture you have is much different in appearance to that because of a different stain. I need to find that purple stain and show you that. Let me see if I can show you a different version of it. Do I have a different version of it? Where's the first slide I showed you? How about this one? This is hold on, let me go down. Well, this is an intriguing slide. Okay, yeah, let's not talk about that. Okay. This is epididymis as well. Much different appearance, right? Stereocilia are actually easier to see here, right? You can see them coming up all over the place, right? And that epithelium is much more distinctive here, right? All right. Anything else on epididymis? So in the area between the cells, in the duct, right? In the area between the duct, what's the one thing that's more common here that actually what's found in this in this so-called interstitial area here that's not found in the interstitial area in the testes. What's it say right there? Smooth muscle, right? Why do you have smooth muscle? Same reason you have smooth muscle for anything, right? In the intestine, what's it for? Push the food down the tube, right? Same thing here, you're trying to push that material down the tube, right? And what you see is, let me see if I can show you what I'm talking about here. Let me go down one more. Look at, let's see. See if I can. Okay, let's do this. Can you see all that? Still a little bit big. Go down one more. Hold on. Oh, that's much better. Okay. When we're talking about the smooth muscle layer and pushing the semen down the tube, right? So this is one seminiferous tube. This is another seminiferous tube. It might be the same one at a different spot. It doesn't really matter. But if you notice it's almost kind of compacted into one little area, though, right? Here's another one here. See how it's in one little area. Here's another one here, it's in one area. Here's another one here, it's in one area. So they're little subdivisions of them that are surrounded by their own little bands of smooth muscle that will compress that whole package. Okay? All right. Let's see, what's next? I haven't seen a vas deferens slide. I'm gonna have to find one. I'm gonna have to dig for that. I don't know where it is. So we'll come back to vas deferens. I need to find my prostate slides, so we can look for those. Is that all we have left in this? I had a penis slide. I saw one of those. I have one of those. So I need prostate slide and vas deferens slides. Time to go hunting. At this. All right. It's not that one. All right, let's see what I can find. Interesting. That's not it. That's not it. Not even the right test? No, that's not the right test. I don't think that's the right test at all. All right. Well, we'll do what we can and then we'll figure out the rest. So I said vas deferens, right? Vas deferens is next. Let me show you a different epididymis slide first. This is an interesting, it's an old one. How about this? This is the one I think I took the picture from your lab manual. Look about right? Or what your picture is in their lab, man? Epididymis?
Yeah, it looks pretty similar.
Pretty clear, right? That's nice and easy to see. That's what makes it a nice slide. All right. Let's look at the vas deferens. What you got?
There's a couple spermatogonia slides on this. They might, I don't know if you wanted to look at those.
I can. Do you want to look at structure of sperm? We can. Sporangia. What are you talking about? Where's spermatogonia? Oh, human sperm sphere.
There's also one that's labeled spermatogonia, if I'm not mistaken. Maybe I misread it.
Sporangia.
I think that's what I misread.
Sporangia is not spermatozoa. Sporangia is plant stuff.
Gotcha.
Sorry, leave it be.
Leave it there? or-
Yeah, that's fine. All right. This is my lowest power. This is your vas deferens here. This is one example of a vas deferens. That's it. That's the entire thing. Now, the vas deferens has the different layers that we think of, right? It's got layers of muscle. You look at the other one, see if it looks, I don't think it's gonna look any different. It's gonna look the same, I think. When you push the other one in, it's probably the same thing though. Yep, same thing. All right, so vas deferens is going to be a little more complex to look at, right? That's not focusing well at all. So your epithelial layer, this is your epithelium here. And then notice that you've got several layers of muscle around the outside, right? Notice that there's an inner layer, and a middle layer, and an outer layer. You got 3 different layers of smooth muscle here. Does everybody see the 3 layers? Maybe. What's the inner one say? Longitudinal. So that means it's going to be cross-cut, right? When you look at it like this, it's going to be cross-cut. So that means this layer here, right? That's your inner layer, right? Your middle layer is going to be this one. And then your outer layer is going to be that one. So 1, 2, 3, right? Now, there are other sections of vas deferens that look better than that, but that is what I have right now. And I'll see what I can do to find a better one. It's going to take a major effort to get this place in any kind of order. All right. So you've got your, what, am I missing anything?
The lamina propria.
So the stratified epithelium, lamina propria, right? Lamina propria is there, you can see it better there. Okay. So here's your epithelium. It goes from here to there, right? Pseudostratified epithelium goes from here to here. This is your lamina propria. See this? Between the epithelium and that first layer there, right? Are you with me? Okay. There's not much to it. It's fairly simple, right? Nice big blood vessel down there, by the way. Those are big red stuff, those blood vessels. So the vas deferens is fairly simplistic. We're going to go ahead and look at the penis, and then I'm going to hunt for prostate gland, which we haven't been able to find. All right, penis cross section. Penis. All right, here we are. So the first thing is you're going to have an epithelium on the outside. Standard epithelium on the outside, right? Stratified squamous, keratinized epithelium. Standard thin skin, right? Then you're gonna have this area called the hypodermis. And if you look, you can see, so here's your epithelium, right? And then you can see that there's a layer that goes down to about here that looks the same, right? So this layer here is your hypodermis. Hypo meaning underneath, right? Underneath the skin, right? Corpora, when you see that word, what's corpora stand for? What does that word mean? What's a corpse? Body, right? Corpse is a body, right? So corpora is a body, a unit, if you will. So when we talk about the corpora, we're talking about this thing in the middle here, like this. This is a corpora, right? You've got two of them. You've got this one, and you've got this one. So one of them is the corpora cavernosa, and one of them is the corpora spongiosa. You want to make bad jokes?
Yes.
Everybody's seen Harry Potter, right? Does it sound like I'm speaking Harry Potter? When I say corpora cavernosa and corpora spongiosa, right? Why? Because they use Latin, right? How dumb is that, right? Just because you speak Latin means it's magic, right? I'll remember that.
It sounds magical.
What's that?
It sounds magical.
Latin? Tell the ancient Romans that. Probably didn't sound magical to their enemies. All right, sorry. So, you've got your devices. Look up the corpora cavernosa and the corpora spongiosa and tell me the difference. You can see them easily enough that there's a difference. Which one's which? So if you Google search corpora spongiosum, what's it say?
Spongy erectile tissue that protects the passage of urine and semen.
Say that louder.
A spongy erectile tissue that protects the passage of urine and semen.
Okay, what's corpora cavernosa say?
A spongy erectile tissue in the penis that's made up of smooth muscle, connective tissue, and hollow spaces called sinus organs.
So one of them has the passage of the fluids out of it, right? One of them does not. Does that make sense? So the spongiosa is the one that has the passage of fluids in it, right? Like maybe through a urethra, which is what this thing is, right? This is the urethra here. So this structure is corpora what? Spongiosum, right? This structure is corpora cavernosum. And what was the first term they used on both of them? Erectile tissue. Did you cover that in any of your classes, how the human penis becomes erect? Well, here's where it gets fun, right? So most mammals actually have a bone in their penis. It's called a baculum. I had a friend in graduate school that studied baculum. or bacilli, right? So most mammals, including things like my dogs, have a bone in their penis. And so that euphemism you know in that you broke your penis can actually be true in those organisms. They can literally break their penis. In humans, we don't have a bone. What happens there? So what do we use to make the penis erect? Blood pressure, blood. So it has to do with the blood flow to the penis and stopping it leaving. So the blood vessels carrying blood in are always open, but the blood vessels going out of the penis can be constricted off and closed off. So that the blood goes in, it doesn't go out, fills up this chamber with blood, makes the penis erect. When that muscle relaxes, blood goes out, deflates, right? Simple enough, right? All right, let's see, what do we got left? Endothelium, we gotta go to the urethra, right? So, what do you see? What is this out here?
Is that albuginea?
Tunica albuginea, right? What is that made out of? What's it look like?
Muscle.
It's a mixture of muscle and connective tissue, a lot of muscle in there, by the way. Urethra, on the other hand, down here in the corpora spongiosum, right? Much different tissue here. It does fill up with blood, not quite as much as that one up there, but primarily for the passage of the semen through the urethra, urine through the urethra, whatever, right? It does have an endothelium, you can see it here, right? Let's look at that a little closer. So what kind of epithelium is that?
Stratified cuboidal.
It's not cuboidal, it's not columnar. Pseudostratified. The nuclei are everywhere, right? All right. Let's see. That's urethra, of course. Anything else over there to cover there? And I need to hunt for a prostate gland. Prostate gland is a very intriguing organ. And the reason that we talk about it in this class is because it's what causes the most problems, right? I probably need to just take these off and see if this is the right test material. I don't think it is. I only have a few prostate gland slides, and they're probably hidden up here. So this is the second test. This is the wrong test material. I'd be willing to bet that I know where those slides are. All right. Where did I see it? I saw it somewhere. Was it on this slide? Hold on. Prostate gland, let me see. I think I have a picture. Is it on this one? Yes, okay, good, all right. So at least we have some prostate gland. This is prostate gland here. That's prostate gland there. Prostate gland has a very relatively distinctive look to it. Looks kind of like the picture, right? Kind of. So you have a lot of smooth muscle around it. A lot of smooth muscle. again, to press that fluid out, right? Prostate gland makes a very basic material to help counteract the acidity of the vagina, help keep the semen alive, right? These are the tubuloacinar glands. It's basically a big gland. It has the fibromuscular stroma. The only thing that I have to show you is this corpora amylacea. Corpora meaning body, right? Amylacea. Anybody know amylase? Amylase? What's amylase? So think, there's not any good ones. Let me get closer. This one doesn't have a lot of it in it. I don't see any real cases of this. There's no real corpora amylacea in here. All right, let's talk about this. So it puts out a very basic material, right? If you have an upset stomach, what do you take for it? What's one of the things you can take for an upset stomach? You said Tums? Okay, what's the liquid version of Tums? Pepto-Bismol, sure, that's fine. Milk of magnesia, same stuff, right? Milk of magnesia is wise, Tums is pink. There's a little more flavoring in Tums, but it's all basically the same thing, right? What happens to any of those if you let them sit out and dry out a little bit too much? Chalky, right? Exactly, they get chalky. They solidify into a solid chalky mass, right? So the prostate gland secretes the same kind of material. So if you let it sit too long in the prostate gland, it will do the same thing, start hardening up, and you end up with these hardened secretion bodies in there. And that's what the corpora amylacea are these hardened bodies in the prostate gland. And as you get older, you get more and more and more of those. And there are, I have a slide somewhere, and I'm gonna try to find it, that shows a young person who has one that's just fine. This is a good example of one that's just fine. There's nothing wrong with this one. And then an older person who has a lot of corpora amylacea in them. And I will try to show you that at some point. Do you have a picture of it here somewhere? Yeah. So, here, this picture? Everybody see this picture? What do you think those big kind of striated looking things are in the middle of the areas there? Those are corpora amylacea. Basically the whole gland is filled up with them, right? So it kind of clogs up the system, right? Does it hurt anything? Can keep the gland from functioning. Can cause pain. Can it be treated? A little bit. Not a lot, a little bit. That's what it is. I will try to find a slide of it. It looks very different when you see it on the slide than it does in that picture, trust me. Anyway, what time is it? How's our time? Been there about an hour. So next week is female reproductive system. I will try to find my prostate slide somewhere in the next week. Maybe I need to spend a day in here sorting slides. Maybe I can get Professor Duncan in here to help me, since he taught this last year, right? I think he lost it. It's all his fault. We're gonna blame it on him.
Can
All right.
Can you point out the basal cells on the surface secretory cells?
Can I do what?
Real quick. The basal cells and the surface secretory cells.
We're That's an ugly slide. Okay, let me find the other one and I'll point it out on that. How about that? Let me find a better slide. Okay? So let me hold you off on that until I find the right slide so I can show it to you correctly. One, then trying to see if I have anything that has it in there. Hang on, I'm still looking. What about the testes? Did I say I was gonna find a different slide of the testes? Let's see this one. How's this one look for testes? Different, right? Different colors, definitely, not pink. What is this one standing for primarily? Nuclei, that's what we're seeing a lot of here, nuclei. Not much else, right? Is that better or worse? Just like many things, when you exclude a lot of stains in there and make things so you can't see them, it makes some things a lot harder to see. But you can do a few things in here. This is seminiferous tubules, right? So what are these things right here? Spermatids, very good, right? What is this nucleus here? That's a Sertoli cell. This is the Sertoli cell here. All right. What's this cell here? Radigonia, right? What about this? Primary, what are these things? Secondaries. I think you got it, right? Doesn't matter what slide we throw up there, right? They're all pretty good. All right, I will look for a prostate slide right now when I have 5 minutes and I don't have anybody to bug me with it. With anything else, I just won't go back to my office. I'll hunt for it. And then next week we'll do female reproductive system. Then you can have a happy Thanksgiving, right? And then come back and cram for 3 days, right? Remember, you have your presentations coming up, right? Okay, that's it. I'll talk to you later. I will put something up on Blackboard. I'm gonna try to do it today, okay?